COVID-19 has caused an unprecedented global health crisis, infecting millions and claiming lives worldwide. The COVID-19 pandemic has reshaped our world and remains one of the most devastating global health crises in modern history. As of March 2026, according to the WHO, more than 779 million cases have been reported worldwide, with an increase of 2,121 new cases in the past weeks. Tragically, over 7 million lives have been lost, with additional 88 death cases reported in the same period. Despite the progress of vaccination campaigns, the continuous spread of variant highlights the urgent need for innovative and sustainable vaccine solutions for this endless infectious disease. Since its first detection in late 2019, SARS-CoV-2 has continued to evolve. The original wild-type strain rapidly gave rise to multiple variants of concern. In late 2020, the Alpha variant emerged in United Kingdom, followed by the Beta variant in South Africa and Gamma variant in Brazil. Soon after, the highly transmissible Delta variant spread worldwide, fueling multiple waves of infection. After that, Omicron variant emerged in 2021, and now its Omicron subvariant BA.3.2 and XFG. This highlights the virus' continued evaluation and persistent challenge it poses to the global health. In the frequency of the color clade also is showing the rapid mutation of the spike protein of SARS-CoV-2. Vaccination is the most reliable solution to combat this infectious disease for now and future as well. Here is the roadmap of developing the pan-CoVax multi-epitope vaccine. Pan-CoVax is a multi-epitope recombinant subunit vaccine. Using advanced bioinformatic tools, epitopes are predicted, screened, and incorporated into recombinant vaccine construct. Following synthesis, protein expression and purification are carried out to obtain the recombinant protein for vaccine formulation. The vaccine is then combined with suitable adjuvants and subjected to quality assessment. To evaluate safetiness and efficacy of the formulated vaccine is further tested in mice model. The preclinical study of pan-CoVax vaccine was done in BIICC mice. The preclinical study initiated with the vaccine formulation, where the purified protein will be formulated with appropriate adjuvant and then will be administered to the mice model. The preclinical study of pan-CoVax in mice model provided valuable insights into the effective dose and, most importantly, the immune responses elicited by the vaccine. Let's hear the uniqueness and novelty of the pan-CoVax subunit vaccine. This vaccine uniqueness is its design strategy. Unlike conventional vaccines, it also integrates both humoral and cellular immune response. By focusing on conserved epitope in designing the vaccine, it provides a multivariant coverage and a broad spectrum of protection against SARS-CoV-2 and its variants. So the uniqueness summarized point of this pan-CoVax vaccine is multivariant coverage, multi-epitope strategy, cold chain friendly, cost-effective designing, and low risk of ADE and rectogenicity. Let's hear the expert's opinion about this vaccine platform.
The COVID-19 pandemic has demonstrated the urgent need for vaccines that provide broader and long-lasting protection against evolving coronavirus variants. A multi-epitope of pan-coronavirus subunit vaccine is a promising strategy designed to address this challenge. This vaccine combines multiple conserved epitopes from different coronaviruses to stimulate strong humoral and cellular immune responses. Unlike conventional vaccines that target only a single strain, this approach offers a broader cross-protection against current and potentially future coronavirus variants. One of its major strengths is safety and precision. As a subunit vaccine, it contains only selected antigenic components, reducing the risk of adverse effects while maintaining effective immune activation. In addition, immunoinformatics also helps to identify highly conserved epitopes, improving vaccine effectiveness across diverse populations. The potential impact of this vaccine is highly significant. It could enhance global pandemic preparedness, reduce the spread of emerging coronavirus variants, and contribute to the development of a universal coronavirus immunity.
In this project we have developed, we targeted SARS-CoV-2 epitopes that covers all the variants. So the antigens for this are screened in silico using bioinformatics pipeline. The epitope designs are novel and has been filed for a patent. One of the principal strengths of this project is that the vaccine is immunogenic and is able to neutralize the SARS-CoV-2 receptor, and it is found to be non-toxigenic in a GLP toxicology studies. So one of the very important aspects of this project is that we can modify the epitopes while the project was going on. So we are able to update the epitopes when the variants were emerging. So basically we just need to change the amino acid sequence in the epitope. One of the important safety aspects of the study is we don't need a pathogenic virus to develop this vaccine. We just use a sequence to develop this vaccine. And in future pandemic preparedness or diseases preparedness, this is a very good platform technology of developing an epitope-based vaccine. We just need the DNA or amino acid sequence for this pathogen. This innovative approach of developing vaccines is a promising vaccine technology in the advancement of next generation vaccinology and global pandemic preparedness. Thank you.
The pan-COVAX multi-epitope subunit vaccine has reached a technology readiness level of 5, having successfully complete design, synthesis, basic evaluation, and prototype validation. Studies conducted include vaccine design, formulation, stability assessment, and immunogenicity analysis. This vaccine development project is supported by the Strategic Research Fund from MOSTI. Currently, this vaccine has completed the GLP study. Patent has been filed for the designed epitope. Here is the certificate of filing. In conclusion, the development of multi-epitope pan-coronavirus vaccine represents a promising and innovative strategy to address the continuous emergence of SARS-CoV-2. By using the strategy of designing a vaccine using a conserved region epitopes, it's really give a potential to create a vaccine which have a broader immunity response against the SARS-CoV-2 variant sequence as well as the cross-protective immune response against the pathogens. And this project is very promising. This is because this platform can be serve as a base platform to design the vaccine not only against SARS-CoV-2 and the future emerging variants. It also can be a good platform to design the future preparations, zoonotic variants, or any other infectious diseases as well. For example, Ebola, and then the current variant, the Hata virus as well. So we are strongly suggesting that this particular vaccine designing platform is promising, and it is also in the case of future perspective, it can be a promising platform to design a vaccine against all the infectious virus. So let's come into the AI-driven strategy to design the vaccine against all the infectious diseases. Thank you.